Hi, welcome back to another lesson. We are going to go over DNA really quick. And for this process, it's going to be pretty big for the test. The main piece of information that you need to know is what DNA is, where it's located, and the base pairs. If you can get those down, you'll be okay for this portion, although it is all over the place. So, knowing them is of pretty vital importance. So, DNA is found in the nucleus and it stays in the nucleus. Um, and so, because it has to stay in the nucleus, that's why you have the process of transcription. Transcription is the process of converting DNA into RNA. Um, so, we can actually write that down. Let's see if we get this transcription, which is turning DNA into a specific kind of RNA, which is called mRNA. Okay, so transcription turning DNA into mRNA. Now, the basis for DNA: ATCG, adenine, thymine, cytosine, guanine. These four bases are used to construct the code for making proteins. That is the purpose of DNA. Instructions for protein shape. Okay. Now, if you go back to when we were talking about enzymes, enzymes have a specific shape and that gives them their function. The same thing is happening for proteins. So, proteins have specific shapes and orders. And if that shape changes, their function changes. And DNA has all of the instructions for all the proteins that your body is going to need in what shapes they need to be in. These shapes are determined by sequences of these bases, these four bases that we talked about before. So these four bases can be put into orders of three bases at a time, known as a codon. And these are going to let the body know the order. So codons placed in a row will tell, okay, this coding is this coding, or sorry, this protein is this, and the specific shape based off of that order. So these codons determine the protein shape, and there are three organized sets of bases. So each DNA molecule is actually going to be two split sides that are going to be twisted together in the form of what's called a double helix. And on each side, the base pairs have a paired molecule that can only go with it. So A can only go with T, and C can only go with G. So this is known as adenine and thymine, and then cytosine and guanine. They go together. So these are purines and pyrines. All right, they always go together. This process of replicating the DNA, so getting all the chromosomes to have a copy, is going to happen during the synthesis phase, the S phase of the mitosis and meiosis. It happens in mitosis and meiosis. So replication of DNA during synthesis phase happens for mitosis and meiosis. It is for both. That is also going to be on the test. So remember that. Interphase occurs for both mitosis and meiosis, not just mitosis. Okay? Because remember, meiosis still needs that copy in order to split twice, and it just isn't always shown. Okay. So in this, we get the three base pairs, the connections, 1 to 20 amino acids. So these codons can code for 20 different amino acids that can be used for different functions. Some of them that are repeating. You don't need to remember any of the purposes or the amino acids. You don't need to know the codons. For example, AUG being start codon. You don't need to know any of that. You just need to know this information here. So DNA to RNA, and their purpose is the instruction of protein shape, and you need to know the bases. So 1, 2, 3, 4. Know each of the bases, what they are, and which base pair do they match up with. That is what the test is going to consist of. So if you can remember that, you'll be good to go. All right. Also, the name is deoxyribonucleic acid. It is one of the two nucleic acids. So there's deoxyribonucleic acid And then there's ribonucleic acid. Why is it called deoxyribonucleic acid? It's because of the structure of DNA, which is I'm going to show here. That will also be on the test. Starts off with phosphate, and then you have your sugar. In this case, it is deoxyribose, and that is where the name comes from. So deoxyribose, and then you have your base pair, and that is the shape of DNA. Now, this will be on the test. The structure will be on the test. You'll need to know which sugar goes into DNA and which base pair will accommodate it. So in this case, we know that AT and CG are for DNA, and so whichever one matches. It might be a U in there, it might be a random letter like a P. That's not for DNA. And if this has an R, if it says ribose, ribose is RNA, so that's wrong as well. So you want the D, and you want a base pair that is for DNA, and that'll be your DNA molecule. Okay, and that's it. That's all you need to know for the test. Hope you have fun. Bye.